Hey everybody, let's try and do this in a little different way today. I'm going to see how this camera works with this particular setup, and we're going to hope for the best. So this is going to be Chapter 9. My first comment to you is this. What follows is going to seem like the most complicated thing that we've done all semester. But if I'm being honest, so the first test, from my perspective, sort of gets your feet wet. The second test is supposed to be the hardest test that I give. And then the third test, people tend to be buckled down and they do vastly better on the third test than on the second. So take that as a, for whatever you will, I suppose, at this stage. But people tend to do well on this. It's going to seem very, very complicated up front. But I assure you, once you sort of get into it, it's not as bad as it looks. All right. So the first time through, it's going to seem like the world is ending. But the second time you hear it and then third, you're going to be like, okay. This isn't bad at all. So just bear with me, all right? It's going to seem bad. It's not going to be as bad as it seems. Yeah, and this is where I tell you about how my public speaking professor in college said never open on an apology. But here we go, all the same. All right, so this is going to be Chapter 9, the muscular system. And full disclosure, it's probably like the second favorite thing that we do from my perspective. Like, I really love doing eyesight and hearing and vision, but this ain't bad. This is good stuff. All right, structural and functional organization of muscles. So your muscles take up a huge amount of your body by weight. Okay, your skin's very large in terms of your body as being a single organ, but your muscles take up a huge amount of the body by weight, basically half. You know that you have about 208 bones in the body. 6? Don't quote me. And then around 600 muscles, okay? 600 muscles. The idea here is that you have far more muscles than you have bones because, think about what your bones can do. You can close your hand. You can open your hand. These require more than one muscle for a single bony structure. So it makes perfect sense that there are way more muscles than there are bones. Let's see, half the body by weight. 3 types being skeletal, cardiac, and smooth. And they are indeed specialized for a single major purpose, and that is to convert chemical energy in the form of ATP into mechanical energy of motion. Now, 3 types of muscle you should be very familiar with at this stage. These are skeletal, cardiac, and smooth muscle. Skeletal muscle is both striated and voluntary. What this means is it has a striped appearance and it is voluntary in activity. So I think flex hand and my hand flexes. Let's just leave the rest of this for later. Cardiac muscle is striated and involuntary. It also has a striated or a striped appearance, but it is involuntary. It contracts without your conscious input, which is a good thing. You don't want to go around having to think about making your heart beat. And then, of course, there is smooth muscle. Smooth muscle is non-striated and involuntary. And what smooth muscle really is involved in is a concept we refer to as peristalsis. Peristalsis is a set of rhythmic contractions. Basically, if you imagine a tube like the intestine, peristaltic motion would cause contraction at one phase and then that contraction to roll down the length of the tube. So you end up with these rhythmic contractions, these peristaltic contractions. And what they really do is they propel things down like the intestinal tract or the ureters or what have you. Okay, it's just sort of what they're involved in. And that's what we're showing you here. So you see this ring closing around a camera, and then that ring progresses down the length of the tube. This is a good example of peristaltic motion. Y'all gotta bear with me, man. This is a totally different setup than I'm used to. All right, characteristics and functions of muscle. So responsiveness is a good way to look at this. Muscles are responsive. When they are exposed to specific chemicals, they are capable of then contracting. For example, when you expose skeletal muscle to its very specific neurotransmitter, this is a chemical released by the nervous system that tells a cell to do something. In this case, causing that muscle to contract. When you expose skeletal muscle to the appropriate neurotransmitter, it causes those muscles to contract by opening gated ion channels. That is how muscles function. Muscles are responsive. They are excitable. Muscles are conductive. You can stimulate a muscle at one end, and the stimulus will flow down the length of that muscle to the opposing end. And that sounds a lot like nervous tissue. We're going to have to talk about the differences between muscle and nervous tissue as we progress. Muscle is extensible. You tend to be able to stretch muscle beyond its resting length, and what you'll find is whenever this happens, it tends to cause the muscle to then automatically contract. You may be familiar with people taking a little hammer and hitting somebody in their, patellar ligament, the old patellar ligament reaction. What they're actually doing is they're stretching the muscle in the quadriceps, and that causes the muscle to automatically contract. Then what do we have? Elasticity, so muscle is capable of recoiling after being stretched. And as we now know, this is not that old of knowledge, but once the muscle is contracted, there are also elastic components internally that force the muscle back out. And then, last but not least, the obvious point here, contractility. That is, the ability to shorten when stimulated. This is unique to muscle. When I ask you the difference between a nerve and a muscle, for muscle, that difference is contractility, that ability to change length. Now, muscles have a variety of functions that you're aware of, moving, holding you up, holding things together, but one you probably don't often think about is the generation of heat. Why, when you get up and have to run from one place to another, or if you walk briskly from one side of a building to the other side of a building, why do you feel hot? Why can you go and watch Green Bay play in the middle of winter and all the football players are pouring sweat? The reason is because when you have physical exertion, muscle contracting, it generates heat. And that's a very important concept. This is why when you lay down to sleep at night, you feel hot because you've been doing all sorts of activities, but then when you wake up in the morning, you feel cold because you've been laying there not flexing your muscles. Ergo, you've lost heat over time. This is an interesting concept. Okay, skeletal muscle. This is an organ comprised of skeletal muscle tissue, blood vessels, nerves, and connective tissue. Now, let me explain what you're looking at here. What we have is skeletal muscle with the fibers running lengthwise, and you're looking at a whole long skeletal muscle fiber. And then here, what you're looking at is if you took this bit and turned it on end and looked down at it, all the fibers are end on. So imagine if I had a bunch of spaghetti noodles and I stretched them out like this, you would be looking at it like that. And then alternatively, if I turned it end on to you, you'd be looking at it like this. So these would be very deep in there. If you take this and you digest away all the musculature, what you're going to end up with is just the arteries and veins, the arterial network, the capillary beds, and it looks something like this. Okay, the muscle is just covered in arterial networks, capillary beds, arteries, veins, you name it. I mean, they're just packing this muscle, which makes perfect sense. Your muscles have to contract and relax and contract and relax. This requires massive quantities of oxygen and will release massive quantities of waste products. So you have to have your blood vessels intimately in contact with all of these muscle fibers all the way down their length. And further, if you take it end on and digest away all the musculature and leave nothing behind but the arterial networks, you'll see that every single one of these muscle fibers is completely encapsulated in capillaries. Your body gets lots of blood flow to your muscles, and you know how important that is because you can sit down in a chair for the wrong way for a short period of time and your legs don't want to work anymore. You can lay down and fall asleep with your arm under you or something and wake up and your arm's completely numb. When you remove blood flow to the muscles, they stop working very quickly. Okay, they stop working very, very quickly. So it's very important that we keep a lot of blood flow in and around these muscles. Now, the way that we do this is knowing that blood vessels tend to travel through connective tissue, and your muscles are just jam-packed with connective tissue. There are several forms of this, and I'm going to go through all of them with you, in relatively quick fashion. There is fascia, epimysium, paramysium, and endomysium. Now, fascia is a connective tissue covering that goes around whole muscle groups. Like your quadriceps in your leg, your quadriceps are made up of 4 muscles, and they're surrounded by a single layer of fascia. Fascia surrounds whole muscle groups. Then, underneath the fascia, on the muscle, each individual muscle will have what's called epimysium. Epimysium. That is the outer muscle covering. Outer, anything myo is muscle. Outer muscle covering. Now, this is a fibrous connective tissue sheath around the whole muscle, and those fibers will penetrate into the muscle. And as they penetrate in, the outer fibers, once they get inside, you would call that the perimysium. Now, let me explain. First things first, we really need to talk about the structure of a given muscle. You have the muscle. That muscle is broken down into little groups called the fascicles. You can see fascicles here. Fascicle, fascicle, fascicle. And inside of each of those fascicles, there will be a single muscle, or let me rephrase. Inside each fascicle, there will be many muscle fibers, or what we'll just say are muscle cells. Cell. Big, long, multinucleated, in this case, striated, voluntary musculature. And for me, man, it looks a lot like bone. Like bone is made up of these osteons, these individual units, and muscle is made up of these individual units that we call fascicles. So, epimysium surrounds the whole muscle. Perimysium is the connective tissue that surrounds each individual fascicle. And if perimysium surrounds the fascicles, then endomysium surrounds each individual muscle fiber. Now, let me make darn sure that we are together on this. The outer muscle covering is epimysium. It surrounds the whole muscle. The same fibers just penetrate into the muscle and surround the fascicles. And at that stage, we call it perimysium. And then penetrating in amongst the muscle cells in the fascicle, we call this endomysium. It is the same set of fibers, all right? It's the same set of fibers. We just call it something different depending upon where it is. And one of the things that I love pointing out to people, if you remember back to bone, and we talked about how bone has an outer covering, a periosteum, connected with Sharpie's fibers, and tendons and ligaments connect to that periosteum. The reality is that all of this fiber inside of that muscle, the fiber amount stays the same, and there's less and less muscle and less muscle, and eventually there's nothing but the fibers that make up all of this, and that's what becomes a tendon. And then that becomes one with the perimysium on the bone, and that's how the tendons connect to the bones. Now, I'm not going to put that on your test necessarily, but I think that's a neat way to view this. You got to think of like the middle of your bicep having a lot of connective tissue in it, and that connective tissue bears the arteries and veins that feed the muscle. But then as you go towards the end of that muscle, you end up with these tendons, and the tendon is just a continuation of all the fibers from the middle of the muscle all the way in, connecting to the bone as a tendon, connecting to the periosteum, connected to the bone via Sharpie's fibers. It's just a neat thing to realize how it's all truly organized. Now, muscles can connect to bone in a bunch of different ways. There's what's called indirect muscular attachment, and also what's referred to as direct or fleshy muscular attachment. We're going to keep this real simple. Indirect muscular attachment is the utilization of tendons, whereas direct muscular attachment does not require a tendon. There are good examples of this. So, I have, apparently got something on my hand from this table. I have a bunch of tendons that run through my wrist here. Where are the muscles that power your hand? They're actually way down here, okay? The muscles here, you can see I can push against this and I can move my fingers, and you can see the musculature moving around in my lower arm here. So they are connected. They have tendons that run up through my wrist, through what's called my carpal tunnel, up all the way to the tips of my fingers, and allow me to run my fingers by using the muscles down here. That is an indirect form of muscular attachment. By comparison, my thumb will demonstrate a direct or fleshy attachment. This is going to be a case where the muscle is on the bone, man. It is attached to it. There's no tendon in between. So you have muscle embedded in here, and it connects to the thumb. That muscle is on the bone, direct attachment. You can see it here as well. The brachialis has no tendon connecting it to the humerus. They are attached to one another. The fibers from the perimysium, endomysium, and which one? Where did I start? The other one, I guess I started at the inside, the epimysium. All those fibers, they just terminate directly on the bone and attach to the bone in that fashion. Your shoulder muscles do the same thing, all this fixation muscles. Now, man, let me tell you something. You need to pause this video All right, you need to pause this freaking video. If you want to learn something, pause the video and open up this one. This is an amazing video of a hardcore, real deal professional butcher taking down a side of beef. Just watch it for like 10 or 15 minutes. You'll see tendons, you'll see fascia, you'll see connective tissue of various formats like fat and how they all attach to one another. You'll see how the muscles just come right out and separate from each other. You will learn so much from this. So pause the video, go give it a watch. You can watch you can go watch Yo-Yo Ma play his instrument. You can go and watch, better idea. Go watch Wynton Marsalis, one of the best trumpet players of all time, playing the Carnival of Venice. It'll blow your mind. You can watch this guy break down a side of beef, and it's the same level of mastery. So give it a try. You'll learn a lot from it. All right, now, YouTube is also your friend. I have linked you to the 3 videos that you need to know as we move forward. Not a terrible idea to pause this video and watch these 3 videos. This one's 3 minutes, this one's 2 minutes, this one's 3 minutes. That is not a lot of time. And you're going to be super confused by it immediately. And then you're going to come back here and listen to me go over it. And it's going to, you'll be like, oh, okay, well, I remember parts of that. And then when I get done, you can watch these again. And chances are you're going to know it pretty good. All right? So go give those a watch. Not a terrible idea. I've linked you to them. It's not hard. Now, here we go. Let's have this conversation. This is where things get a little bit interesting, folks. Skeletal muscle cells, man, let me tell you, they're just different. They're just a little bit different from every other muscle in your body, from every other cell in your body. They're unique. And because they're so unique, they get their own sort of tagline, if you will. Anything sarco as a root. Anything sarco, we're talking muscle, typically skeletal. So for instance, well, hang on, let's just run through. Let's not get ahead of ourselves. Skeletal muscle cells are enormous, way bigger than typical cells. Like you'll have a single skeletal muscle cell running from here to there, one big long cell, microscopic in diameter, but a big long cell. You can have single skeletal muscle cells that are yay long, okay, up to 30 centimeters. These are very, very large, like those in your legs. And because they are so long, they have to be multinucleate. One nucleus just can't keep up with the protein demands and can't get protein where it needs to be. So you have multiple nuclei all throughout the length of skeletal muscle. Now, a few modifications. Skeletal muscle cells have what's called a sarcolemma. That would be the plasma membrane of the muscle cell. Now, all cells have a plasma membrane, but skeletal muscle cells have a weird plasma membrane called the sarcolemma. It is so different that it gets its own name. It's no longer a plasma membrane, it's the sarcolemma. Skeletal muscle cells have cytoplasm just like every other cell in your body, but it's so different that we don't call it the cytoplasm, we call it the sarcoplasm. Now, how is it different? It contains a lot of glycosomes. These are stored glycogen that can break down into glucose very quickly to provide energy for the cell. If you've got a lot of glucose, you're going to need a lot of myoglobin. Myoglobin is a protein network, a lot like hemoglobin. It basically stores oxygen, for your cells. So this is going to be an emergency oxygen storage. This is an emergency sugar storage. And then you have piles of mitochondria inside of these muscles. I mean, these are just packed with mitochondria because they got to burn through that sugar using that oxygen to make copious quantities of ATP so that the muscles can run in emergency situations. And just as an aside, if you go to Publix and you look at the meat counter and you look at the steaks and they look red, the reason they are red isn't because of blood. That's foolishness. Blood gives meat a terrible taste. They drain the blood out almost immediately when they kill the cow. The reason the muscle is red is because the myoglobin. Okay, myoglobin, this protein network, is also iron-based, and it gives that muscle its reddish appearance. When you cook a steak and it turns kind of brown, what you're doing is you're breaking up the myoglobin and it changes color. You get this dark, rusty color initially. Then if you cook it long enough, you could just wear it on your shoe. Anyway, let me not talk about how you should cook your steaks. That's a story for another day. Now, there are 3 highly modified structures in muscle that you need to be intimately familiar with. These are the myofibrils, and boy, let me tell you, whew, it's a long story, okay? Myofibrils, complicated. Then there is the sarcoplasmic reticulum. Sarcoplasmic reticulum sounds like endoplasmic reticulum. The sarcoplasmic reticulum is basically the smooth ER of the cell, but this case being a muscle cell. So we don't call it the smooth ER, we call it the sarcoplasmic reticulum. And the reason for this is because it does a job that it doesn't do anywhere else. The sarcoplasmic reticulum in a muscle cell just stores calcium. Lots of it. Stores lots of calcium, man. Then we have these very unique structures called the T-tubules. T-tubules are parts of the sarcolemma, and basically what they do is, instead of, like on a given cell, a cell will have a surrounding membrane on it. And so do muscle cells. They've got a sarcolemma. It's a covering on the outside of the cell, but skeletal muscle cells have T-tubules. These are areas where the sarcolemma penetrates into the cell and goes deep all the way to the core of the cell. So the outer membrane has these areas that are like tubes that penetrate all the way to the core of the cell. Those are T-tubules, membrane invaginations. All right, let's talk about some freaking myofibrils. So the myofibrils, quite simplistically, are actin and myosin. They are actin and myosin are the contractile units of a muscle. Well, let me rephrase that. They are the contractile proteins of a muscle. The contractile unit of a muscle is called something else on the next slide. Now, the actin and myosin give rise to the unique striated appearance of muscle. All right, what we have are thin fibers called actin molecules, actin fibers. They're the blue ones here. And then the thick fibers called myosin fibers, which are the red ones here. And it gives rise to this unique striped appearance. The stripes are bands of actin and a band of myosin, and a band of actin and a band of myosin, and a band of actin and a band of myosin. It's very simple, okay? That is what we are dealing with here, these myofibrils. Now, I will point out the following. If you look at this, we have these areas where the myosin, these areas where the actin molecules come together. These are referred to as the Z discs. You don't need to know the rest of this, but that's a Z disc right there. Okay, those are Z discs. The area between a Z disc, so here's one, there's 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 one. These are called the sarcomeres. Let me say that again. The area between 2 Z discs is referred to as being a sarcomere, and a sarcomere is one functional unit of a muscle cell. Yeah. Boy, let me tell you, there is so much more we could talk about here, but it's freaking online, and I don't want to take you too deep, so let's just leave it there. The area between 2 Z discs, that, ladies and gentlemen, that is a sarcomere. All right, now let's go into our actin and myosin. These are referred to as being the thick filaments and the thin filaments. Thick filaments are myosin. And myosin is like a big dumb molecule, okay? It's very simple. Myosin is just myosin. A myosin strand, a thick filament, will just be a whole bunch of individual myosin molecules all bound together. Literally hundreds of them. Hundreds of myosin molecules all bound to one another form what's referred to as a thick filament. And these are special because they bear an enzyme called ATPase. ATPase is very important for converting... oh, geez, I'm totally blanked here. Let me try that again without actually looking at the screen. No. Dyslexia. Myosin can break down ATP. There we go. So myosin is the ability to cause ATP to impart energy, and that allows myosin to move. These little heads here, they're like little grabby hands. So what they can do is they sit in a fully energized state, ready to do their thing, and the moment actin becomes available, they grab actin and pull. And then they're stuck. And what you have to do is you have to burn ATP to break them loose and re-energize them. And I forget where I was. So what you have to think about is this. You got to imagine that actin and myosin is kind of like a person climbing a ladder. The ladder does nothing to force the person to climb it. The ladder is simply a substrate upon which climbing happens. That's actin. Okay, actin is a ladder. Myosin does the work. It's got these grabby hands. It's got ATPase. It's capable of pulling against myosin and causing movement within the muscle. So when you see these sarcomeres shortening, what you're looking at there is the myosin heads grabbing actin and pulling. Okay, they have little grabby hands. They have ATPase. And then there is myosin. Wait, did I totally screw that up? Myosin has the grabby hands that does the pulling. Actin, man, let me tell you, when it's two things, the dyslexia hits me hard. All right, thin filaments, here we go. Thin filaments, this is referring to actin. Now, actin is not actin. The reality is the thin filament is referred to as being an actin complex. It's multiple things. So you have actin subunits, you've got troponin, and you have a strand unit molecule called tropomyosin. And it's sort of just a beautiful, elegant story as to how all this fits. Actin is going to bear these unique binding sites for myosin. And if the binding sites for actin are free, man, let me tell you, myosin is going to be grabbing and pulling. Anytime actin binding sites are open, myosin grabs and pulls. And that's just how this works. So at this moment, the muscles in my hand, they are not contracting. But yet there's actin and myosin all in there, all around my hand there. So why aren't the muscles contracting? And the answer to that question is that the actin binding sites are blocked. The actin binding sites are blocked almost all the time. The only time that my hand will contract is when I tell my hand to reveal the actin binding sites, and my hand flexes. The muscles contract. And when I tell my hand to reblock the binding sites on actin, my hand relaxes again. So the question becomes, how do we do it? How do we convince the hand to block the binding sites? How do we convince the hand to reveal the binding sites? Actin, let me rephrase, myosin is always ready. It's just a matter of if actin is available. Okay? It's like any relationship, really. There's always the one that's like super into it and the one that might not be as into it. Okay? And that is how this works. So, the way in which we block the actin binding sites is going to involve this troponin complex and this strand called tropomyosin. Tropomyosin is not complicated. It's just a big long strand and it blocks the actin binding sites. When tropomyosin blocks the binding sites, your muscles are relaxed. They can't contract. Troponin holds the tropomyosin molecule, all right? It's like this strand here. Troponin holds it over the actin binding sites so that the muscle is not contracting. Troponin has to be convinced to pull that tropomyosin molecule out of the way and reveal the actin binding sites so that myosin can grab and pull. And how do we convince troponin to reveal the actin binding sites? How do we convince troponin to pull tropomyosin out of the way and reveal the binding sites so that myosin can grab and pull? We do this using calcium. Now, does that sound familiar? Where do we store a lot of calcium? We store it in the sarcoplasmic reticulum. And what does that calcium do? It binds to troponin, and that causes troponin. See, calcium is an ion, and when you bind an ion to troponin, what it does is it changes the molecular conformation. That means the shape. So troponin sits like this, and then when you bind calcium to it, it leans out of the way. And when it leans out of the way, it pulls tropomyosin off of the actin binding sites and allows myosin to grab and pull. Now then, let me lay that on you one more time. Myosin is a thick filament. It's always ready to do its thing. It's stored at high energy all the time. It's always there, ready to grab a hold of actin and pull. But actin is not always available because it's oftentimes blocked by tropomyosin, which is held in place by troponin. But if you expose troponin to calcium, it will change shape and pull tropomyosin out of the way, revealing the actin binding sites and causing the muscle to contract. So, my hand, it's sitting here at rest. And I send a nervous transmission down the length of my arm through part of the nerve called an axon. It reaches the muscle, releases a neurotransmitter, which binds to the muscle and convinces your sarcoplasmic reticulums to release calcium. That calcium binds to troponin, pulling tropomyosin out of the way, leading to my hand contracting. So what's happening there is calcium gets released internally, it binds to troponin, pulls tropomyosin out of the way, my hand contracts. How does my hand relax? You get the calcium out of there, the troponin goes back to its resting state, blocks the actin binding sites with tropomyosin, my hand relaxes. That's how that works, man. That's how that works. It's all about the interaction of actin and myosin. It's all about the way in which troponin holds tropomyosin to block the actin binding sites. Yeah, that's great. All right, next, let's go into a little more detail here. So we got our sarcoplasmic reticulum. The sarcoplasmic reticulum, which stores calcium, I mean, it is this crazy branching network. When you think about a given animal cell and you look at the, what do you call it, the endoplasmic reticulum, the smooth ER is what this really is, it's smooth ER. You think of something quite small, but in the sarcoplasmic reticulum, in muscle cells, the sarcoplasmic reticulum is massive, and it's super branching, and it just completely surrounds the muscles. And it's packed with calcium. Now, here is something that students screw up all the time, and that is you need a knowledge of the terminal cisternae, or the terminal cisterns, okay? Terminal cisternae are the bulbous ends of the sarcoplasmic reticulum, and what makes the terminal cisternae important is that they bind to the T-tubules. So we got T-tubules, which are invaginations from the cell membrane. They are coming in and surrounding the muscle cells, specifically connecting to the sarcoplasmic reticulum's terminal cisternae, which are here. So all this blue is sarcoplasmic reticulum. The bulbous end where the T-tubule is, that's a terminal cistern. Okay, terminal cisternae. So we got our T-tubules, we got our terminal cisternae, we know it's full of calcium, and this is the cell membrane. The way all this fits together is when you stimulate the outside of that muscle cell with the neurotransmitter, it causes a stimulus that travels down the length of the surface of that cell. And in a regular cell, it will just go down the surface and be done with it. But skeletal muscle cells have T-tubules. So when the stimulus goes down the length of that muscle cell, it travels all the way to the core. The stimulus from the outside travels all the way to the core of that muscle cell, where it stimulates all of these terminal cisternae. They let out lots and lots of calcium. That calcium binds to troponin, which pulls tropomyosin out of the way, revealing the actin binding sites so that myosin can grab and pull. All right? You should be worried right now. At this moment, you should be like, holy cow, this is complicated. But the more we do it, the easier it's going to get, okay? Good. So we know about the sarcoplasmic reticulum. We know that it stores calcium. We know about the T-tubules. We know that they are invaginations off the cell membrane and that they go to the core of the cell. So my question to you is, if I stimulate the outer surface of a muscle cell, does the stimulus stay on the surface? And the answer is no, it doesn't. It goes all the way to the core. Yeah, man, and that's what this is showing you. You got your sarcoplasmic reticulum, you got these little holes of the sarcolemma. These are openings into the T-tubules, so those T-tubules get stimulated. I set a stimulus off here, it flows down. The T-tubules stimulate the sarcoplasmic reticulum, specifically the terminal cisternae of the sarcoplasmic reticulums. When the terminal cisternae are stimulated, they vent calcium. That calcium binds to troponin, pulls tropomyosin out of the way, revealing the actin binding sites and leading to contraction. And that, folks, brings us up to the neuromuscular junction. We'll oftentimes call this a synapse. Not entirely correct, but we oftentimes call it a synapse. This is the neuromuscular junction. So what the heck is a neuromuscular junction? Well, that's an area where there are three parts. There's an axon terminal, a synaptic cleft, and a motor end plate. Now, what the hell are all these bits? Well, the axon terminal is quite simply the end of an axon. What's the terminal end of an axon called? It's called an axon terminal. Nothing complicated, okay? So it's going to come down, the axon's going to come down from the brain, and it's going to connect to the muscle at an area called the motor end plate. The motor end plate, as shown here, is a area of the muscle cell that is receptive to neurotransmitters. Nothing fancy. Okay, it's just receptive to neurotransmitters. So if neurotransmitters get released from the axon terminal, they have to cross a little gap called the synaptic cleft. They bind to special proteins at the motor end plate. It's receptive, and that's what gives off your stimulus that travels down the length of the muscle cell. Does the stimulus stay on the surface? No, it goes down deep using the T-tubules. T-tubules, which are connected to the sarcoplasmic reticulum, in particular the terminal cisternae of the sarcoplasmic reticulum, which then vents calcium, which binds to troponin, pulls tropomyosin out of the way, revealing the actin binding sites, and allowing myosin to grab and pull. The muscle contracts. Yes, good. I'm going to do this to you a million times, so just prepare yourselves. Now, we know that we've got our axon terminal. There's a gap called the synaptic cleft, and that's going to terminate on the muscle cell in an area called the motor end plate. Now, inside of the axon terminal, there are little vesicles called synaptic vesicles, and they store a neurotransmitter. Now, in skeletal muscle, the neurotransmitter of choice, what we use, and you need to know this, is called acetylcholine, or ACH for short, acetylcholine. Acetylcholine is a neurotransmitter. When it's vented via exocytosis from the axon terminal, it crosses the synaptic cleft, binds to protein-based gated ion channels that then open up, and this is all caused by acetylcholine. Okay, these are acetylcholine receptors. This is a real thing. You can see it. Feel free to read all this. Oh, shoot. I thought I had 2 slides. Let's just do it on one then. I'm going to walk you all the way through this. I probably will do it twice, and then you should pause it and rewind it and watch it again. Then you should pause it and rewind it and watch it a 3rd time. Here's how muscles flex. This is how your brain communicates with your muscle to lead to muscle contraction. When I think flex bicep, I send a message down the length of a neuron in my body. Now, what's the long part that conveys messages called? They're axons. I'm sending a message down the length of an axon out to my bicep. And when the stimulus reaches the end of the axon, an area we call the axon terminal, the last little receptors on here are calcium receptors. Now, this is different calcium than you're thinking about. This is different calcium. It pops open these calcium ion channels, and calcium enters into the axon terminal. This causes an internal change to the axon terminal, and it basically stimulates exocytosis. So, message comes down the length of the axon, hits the axon terminal, pops open some calcium channels, which force exocytosis of acetylcholine, the neurotransmitter inside of your axon terminals. When acetylcholine is released via exocytosis, it crosses the synaptic cleft and binds to the motor end plate. When acetylcholine binds to the motor end plate, it pops open some gated ion channels. And now, this is so very important. You need to know this. When the gated ion channels open, they allow the influx of sodium and the release of potassium. All right? Sodium goes in, potassium goes out. You may remember that at some stage I've mentioned that you have sodium-potassium pumps that make sodium and potassium be at opposite ends of the cell. This is why. We want them to be polarized, and then we want to cause the cells to become depolarized, and that stimulates them to do some sort of action for us. Okay? In this case, when acetylcholine crosses the synaptic cleft and binds to the motor end plate at these gated ion channels, they open, sodium enters the cell, and potassium leaves. What this does is it causes a local charge variation around the motor end plate. It causes a local charge variation around the motor end plate called an end plate potential. And what that's going to do is it's going to trigger what's referred to as an action potential. And that action potential is the stimulus running down the length of that muscle cell, which leads to flexion. So, let me walk you through this. Let me see what the next slide is. Yeah, okay. So what we have is our axon terminal. A message comes down the length of the axon, hits the axon terminal. That's calcium into the axon terminal. This causes the exocytosis of these synaptic vesicles containing acetylcholine. Acetylcholine binds to the motor end plate after it crosses the synaptic cleft, When acetylcholine binds to these unique receptors, these are gated ion channels, they allow sodium to come into the muscle cell and potassium to leave. This triggers an in-plate potential, which then propagates over the length of the cell, called an action potential. Basically, the entire length of the muscle cell is just covered in gated ion channels. And when you stimulate a few of these, it just cracks them all open. It's like a freaking avalanche coming down a mountain. Once it starts, it's heading to the ground. Okay? And basically what's going to happen here is you're going to have a wave of gated ion channels just go pop, 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 all the way down the length of this muscle cell. If you remember, back at the beginning, we said that muscle cells are conductive. That means that you can have a wave of depolarization flow down the length of that muscle cell. On the surface, these are surface proteins. Pop, 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 sodium coming in, potassium going out. That is just how this works. Okay, it's all about the movements of sodium and potassium. A wave, this is important, you need to know this, a wave of depolarization followed by a wave of repolarization to put things back where they're supposed to be. Now, does the stimulus here stay on the surface? No, it doesn't. It travels in deep to the core of that cell, right? Deep, deep, deep to the core. Did I already miss it? I guess I did. Here we go. It travels deep to the core of the cell using the T-tubules, which stimulate the terminal cisternae of the Sarcoplasmic Reticulum, which vent calcium. That calcium binds to troponin, which pulls tropomyosin out of the way, revealing the actin binding sites, and myosin, which is stored at high energy, will grab and pull as long as there is ATP in the system. This will happen as long as there is ATP in the system. And that's what I'm trying to show you here. The release of acetylcholine binding to the motor end plate causes a stimulus which propagates over the length of the muscle cell. It doesn't stay on the surface. It travels deep via T-tubules, stimulating calcium to be released from the terminal cisternae of the sarcoplasmic reticulum. That calcium binds to troponin, pulling tropomyosin out of the way, revealing the actin binding sites, allowing for what we call the cross-bridge cycle. This is a term you need to know. The cross-bridge cycle refers to how ATP, actin, myosin, all of these come together and cause muscle contraction. The term is a cross-bridge cycle. Now, I'm going to tell you about one more term here, and that is calsequestrin. Calsequestrin is a unique protein that you need to be aware of. You see, calcium is inherently difficult for your body to handle. So if you're going to store large amounts of calcium in a single area and prevent those from just diffusing all over creation, you have to have something special to hold them, and that protein is called calsequestrin. So you have calsequestrin in the terminal cisternae of your sarcoplasmic reticulum. That's how you're capable of storing intercellular calcium at concentration without it just diffusing away. You have to have this protein to do it, calsequestrin. All right, here's the cross-bridge cycle. Now, I am not going to go into a huge amount of detail here. In fact, I think I'm going to keep it very simple. For your muscles to contract, calcium has to bind to troponin. Troponin has to pull tropomyosin out of the way, and that's got to reveal the actin binding sites. Now, what follows is what you need to remember. Myosin, stored at high energy, right, always stored at high energy, is going to grab actin and attach to it chemically. And then it's going to impart energy. It's going to pull. And now it's stuck there, folks. It is stuck there. It ain't going nowhere. That muscle ain't shortening anymore. My Alabama's coming out because it's like late at night. All right. It's stuck there. And the only thing that breaks it loose is ATP. So when ATP binds to the myosin head, it breaks loose. And the ATP imparts energy to it. It goes from ATP to ADP. And what will happen is, when you impart energy, it re-energizes and goes back to its high energy state. And then if the actin binding sites are still free, it grabs and pulls. And now it's stuck there until ATP binds and disconnects it, re-energizes it, grabs and pulls, stuck there. ATP breaks it loose, re-energizes it, grabs and pulls. And the cycle, the cross-bridge cycle, continues and continues and continues until you remove calcium from the system. All right? When we remove calcium from the system, it goes back to its resting state. The ATP binds to myosin, it breaks loose, and then boom, you slip tropomyosin around the actin subunits, and you're stopping the contraction. You can go back to resting. Tension length curves, this is very simple. The idea is that if your muscle is at just the right length, it's going to have maximum strength. What you want is you want your myosin molecules to be at optimum overlap with actin. Imagine you're going to climb a ladder, but you have to reach up and grab the ladder to try to climb it. If you can't get your hands and feet on there, you're going to have a hard time climbing it. The idea is this. If you've ever seen somebody grab a pull-up bar and just hang from it, they are never pulling themselves up that bar. Because when you stretch the muscle out, there's no overlap between the actin and myosin. So the little grabby hands of myosin can't grab the actin to pull. You get no, what's referred to as tension out of this. You generate no force, no tension. And then by comparison, if the muscle's contracted, I can't really contract it anymore. So once the sarcomeres are completely overlapped, the muscle can't shorten any further. The idea is you want to have optimum overlap. There's an optimal level in this tension length curve where the muscle is at maximum strength. And you have seen it happen. People go to arm wrestle. And you see them start out and they're like even. Okay, everybody's very even. And then one starts to win and the other starts to lose. The one that's winning hits the optimum overlap. The one that's losing is beginning to lose myosin heads off of actin. So bam, they slam the hand down. Pow! The idea is that one of them, the muscle's getting even stronger the more they win, and the other, the more they lose, the weaker the muscle's getting. And you know this. This is how this works. Yeah, that's good enough. Relaxation. You know, this is basically the opposite of what we already did. Man, where do you even start? Oh, there is an important term to be used here. So, yes, if you want to, let me just ignore this. If you want to stop a muscle from contracting, you just kind of work backwards. You think, okay, I'm going to stop releasing acetylcholine. Okay, so I cut off the release of acetylcholine from my nervous tissue, and what that's going to do is there's going to be some acetylcholine stuck in there still that's been released, and it's already in the synaptic cleft. But luckily we have an enzyme called acetylcholinesterase. You need to commit that to memory. You have an enzyme called acetylcholinesterase, and what it's going to do is it's going to break down all the leftover acetylcholine that's stuck there. So you break down all that acetylcholine and it's gone. The reason I can flex my hand very quickly and relax it just as quickly is because we have acetylcholinesterase that breaks down the leftover acetylcholine. Okay, now we break down the acetylcholine that's there. That's going to stop stimulating the sarcolemma. That's going to stop stimulating the T-tubules. That's going to cut off calcium release from the terminal cisternae. So you start pumping calcium back into the terminal cisternae to restore it on calsequestrin. That's going to cause tropomyosin to go back to its resting state and pull, let me rephrase, cause troponin to go back to its resting state and reblock the binding sites with tropomyosin. ATP is going to break the myosin heads loose and they're going to go back to their high energy resting state. Yeah, man, that's how you do this. That's how you relax a muscle. You basically just work backwards. If you understand how you get there, you understand how you stop it. Now, there is a little bit more that I want to talk about. I know we're kind of getting long here, but that's just life. This is a hard chapter. Okay, there's some terms you need to understand. Let me make sure I'm not going to get too far. Yeah, because I want to stop here. There are some terms you need to understand. Those terms are muscle tension and load. Muscle tension is the amount of force that muscles can exert. So my bicep is capable of exerting a certain amount of force. If I lined up barbells from 20 pounds to 100 pounds, somewhere in there, I would run into a barbell that I can't physically lift. That's the maximal amount of tension that my muscle is able to generate. Load is the force exerted by the weight of whatever object it is you're trying to move. All right, so a 20-pound barbell has 20 pounds of load. Here's my glass that I'm drinking water out of so I can sit here and talk for an eternity. This glass weighs about a pound. That would represent about a pound of load. So if I'm holding my glass stationary, that means that I'm exerting about a pound of muscle tension to overcome this pound of load. If the glass starts to fall, that means I'm exerting less tension. If the glass starts to rise, that means I'm exerting more tension. I'm overcoming the load and lifting the cup up. Now, an interesting nuance. How is it that I am capable of cradling a newborn baby in my big strong arms, and alternatively capable of taking a tangerine and squeezing it into paste with my hand. How is it that I can have so much dexterity that I can pick up this pen with great gentility, but alternatively, if I wanted to, man, I could take my thumb and I could pop that sucker right in half? How is that possible? Well, the answer to this is that your muscles are broken down into what we call motor units. Okay, your muscles are broken down into motor units. Whenever I very gently pick up the pen, I'm activating just a few motor units. If I wanted to push against the end of this, I'm activating more motor units. And if I want to break that sucker off, I'm going to activate more motor units until it gets to a point that I have activated all the motor units in my muscle, and it's going to be flexing at maximal tension, max tension. Okay? So your muscles are broken down into little minor subunits called motor units. Some muscles have piles of motor units. Think the ones that allow you this great level of dexterity. And then, like the muscles in your legs, have very few motor units because what they do is a very coarse motion. You don't have as much control over them. Yeah, that'll do. Okay. A given muscle can be stimulated electronically, and we can look at the amount of tension that it generates. If I wanted to, I could take an electrode and attach it to a muscle of yours and send a zap right through it. And when I zap that muscle, what I'm going to see is a chart of tension over time, just like this, exactly like this. This is the amount of tension generated, the amount of force over time. A single muscular twitch, if I zap that muscle, just one zap and call it a day, it's going to look just like this. What you're going to see is a little latent period, right? A little latent period where nothing's happening. Followed by a period of contraction, which is pretty fast. Followed by a period of relaxation, which is much longer. By the way, this is in milliseconds, and it takes 1,000 milliseconds to make 1 second. So this is very quick. Why is there a latent period? Well, if I zap that muscle, what I'm doing is I'm stimulating the outer surface of the muscle. That stimulus has to go deep with the T-tubules. That's got to attach to the Sarcoplasmic Reticulum. Sarcoplasmic Reticulum's got to vent calcium. That calcium's got to bind to troponin. Got to pull trypamyosin out of the way, reveal the active binding sites. Myosin's got to grab and then pull. All that has to happen before we actually get tension. So there's a latent period. A little break when the muscle's gearing up to flex, and then boom, it flexes and generates tension. Very short amount of time to do that. And then the resting state, going back to relaxation. Oh man, that's a long time indeed. Way longer. 5-6 times longer. Why? The answer to that is that it takes a lot longer to put calcium back where it goes than it does to release calcium. It takes a lot longer to let sodium out than it does to use a pump to pump it back in. It takes longer to relax the muscle than it does to contract it. All right? And different muscles in your body do this at different rates, but generally speaking, this is sort of what we're going for. Okay, that's a good general idea of how muscle physiology works for a single muscle fiber. Then we have wave summation. Now, if I take my single twitch, what I can do is, if I've got my electrode in there, I can give you a zap. All right? And what that's going to do is it's going to zap you, twitch. Okay? So there's a stimulus, that's what the S is, and there's your twitch. I can do this 1,000,000 times. Zap ya, twitch. Zap ya, twitch. Zap ya, twitch. And it's going to be identical to this every single time. It's never going to change. Until I start giving you 2 zaps back to back. This is what we're doing. We're causing what's referred to as wave summation. If I give you 2 zaps back to back, you get the first building of tension, and then as the muscle starts to slowly relax, a second stimulus comes along, and it zaps the muscle to a higher level of tension. Basically, it's wave summation. Muscle exposed to 2 stimuli in rapid succession will generate extra tension. The first one, or let me rephrase, the second one builds off the first one. So if I send you a stimulus, you get the bump. And a second stimulus, a second higher bump. And if I get them close enough together, they're going to build higher and higher and higher. And the idea is that if I'm sending piles of stimuli in very quick succession. I'm going to stimulate this wave summation to generate the maximum amount of tension that your muscle can generate. Remember, if I had barbells from 20 to 100, somewhere in there, I would not be able to lift that barbell. There would be a maximal level of tension that my bicep is capable of generating. And I will get there via wave summation. Now, let me check something here. Yeah, okay. So what we have looked at is how a single muscle fiber twitches and how that single muscle fiber can be made to generate more tension by sending more stimuli per unit of time, building one off the other. That is wave summation. And then we need to talk about how your muscle generates tension as a whole. And they do this via recruitment. Remember our motor units? All right. Motor units can be recruited over time to make the muscle flex more strongly. So here is our muscle. If I need to lift something relatively lightweight, like my cup here, my bicep will only activate just a few motor units. Not much. Okay, just a few motor units, not a whole bunch. If I need to lift something heavier, like a 20-pound barbell, I'll activate more motor units. If I've got a 60-pound barbell there and I'm trying to show what a tough guy I am, I could probably lift it. But man, it's going to take everything I've got. You're going to activate every single motor unit in the muscle. Again, this is called recruitment. The concept is that as you generate, or let me rephrase, as you need more and more tension, you activate more of the muscle. So here's the whole muscle. Here you're only activating a few motor units. But as you generate more and more tension, you're activating more and more motor units. That's actually backwards. You generate more tension by activating more motor units. There we go. That's better. There was something else I wanted to say here. All right, if I had a 20-pound barbell, if I held that thing out like this, I could only hold it out for, you know, probably 10 or 15 seconds. I mean, that's a lot of weight to hold straight out from your body, and your muscles would fatigue very quickly and break down, and you wouldn't be able to hold it up. But what about my coffee cup? Coffee cup? No. What about my water glass? Man, I could hold my water glass out here for the duration. I could hold it out there for another 30 minutes, no problem. I mean, it would suck, but I could do it. You know, it ain't that hard. Why is it that I'm capable of holding up a lightweight for a very long period of time, whereas a heavyweight would cause my muscles to fatigue very quickly? The idea is the cup there only activates a couple motor units. And what happens when those get tired? They shut down and rest, and you activate other motor units. What happens when they get tired? They shut down and rest, and then you activate other motor units. The idea is that you're capable of switching these around to keep the muscle fresh. That is how we are capable of holding up weight for a prolonged period of time. The recruitment strategy is that you go to the fresh muscle fibers, and you can keep cycling them over time until you fatigue the muscle completely. Okay, I'm pretty happy. Pretty happy with that. Let's go here. Isotonic versus isometrics. An isotonic contraction comes in two flavors. They're either concentric or eccentric isotonic contractions. Isotonic meaning that the muscle changes length. A concentric contraction is when the muscle shortens, and an eccentric contraction is when I push against my arm and I slowly let my arm straighten. The idea is that I'm contracting the muscle and I'm slowly relaxing it. The muscle is contracting, but it's contracting as it gets longer. Now, I can't push with a muscle. I can only pull. So the idea is that this is putting the brakes on. I'm pushing that arm down, but I'm using that muscle to slow the contractile process, or the movement. I'm using the muscle to slow the movement. And then, of course, there are isometrics. An isometric is when the muscle contracts, but it doesn't change length. I am doing isometrics right now. My head isn't falling over. It's not moving, but it ain't falling over. The muscles in my neck are maintaining a constant level of contraction to hold my head up. Here's my beautiful wife holding up my baby girl when she was tiny. She's doing an isometric contraction. She's just holding that kiddo up. This is when they were supervising me working on a door, and I had to pull up and take a picture because it was just too much cuteness overload. Love those, love my girls. Anyway, moving on. And my boys, but you know how these things go. Moving on. An isometric contraction is when the muscle flexes, but it doesn't change length. This is classic posture, basically. And that brings us to muscle tone. So muscle tone is a concept where the muscles maintain a mildly contracted state all the time. Have you ever watched people go to a gym and they walk in like normal people, but then when they leave, they're acting like they're bodybuilders. They like have this big bulked up look. That's not all in the head. Okay, a lot of that's actually bodily. I would invite you to grab a hold of something small and tightly grip your fist around it for a few seconds. Okay, really like a minute. You just grip it for like a minute and you really hold that thing on there and you're really giving it to it, okay? When you decide to take the pen out and relax your hand's going to want to stay contracted, okay? It's not going to want to sling out easily. Whereas this hand will just hang flat. This one wants to be bent. And the idea is that my brain detects that I was just exerting a lot of force, and perhaps I might do that again. It's like a warning sign, okay? So what the brain does is it leaves, via spinal reflexes, the muscles in a mildly contracted state so they're ready to do work again. This will stabilize joints and help you maintain your posture. So if you go and you work out all the time, you're doing a lot of exercise, your muscles are going to stay in a mildly contracted state. It's the same thing as if you go outside and suntan all the time. You start working in the fields, your skin tone will get darker because you produce melanin as a protective measure. If you work out all the time, your muscles will be in a mild state of what we call tonus. It's a protective measure to stabilize the joints and help keep the muscles prepared to do work because the body thinks more is a-coming. Okay, when you exercise a lot and it builds tone, that's what's happening. And I believe that's all we need to do at this stage. I think I have other recordings for this one, so I'm going to upload those as well. All right, thanks guys. I hope this worked out. Have a good day.